right, hello everyone. I'd like to thank the organizers for giving me this opportunity to present our research today. And this talk is going to be a little different because I am one of those researchers that were talked about in the panel this morning that need to be reined in sometimes. And my goal really is to take advantage of a lot of the technical advances that many of the people in this room have made to try and answer biological questions. And as a neuroscientist, I am interested in the brain. So we'll have just a little bit of biology in the beginning, but then I promise we're going to get to segmentation and quantification. So the way the neurons in your brain talk to one another is through synapses. And synapses are an actual physical structure in 3 dimensions, of course. And here is in panel a single SEM plane showing a synapse in human brain. And right next to it is a schematic of that. So there are basic components of a synapse that need to be there for the neurons to talk to one another. They include the presynaptic bouton shown in pink. That bouton is filled with vesicles. Those vesicles have neurotransmitters in them like dopamine or glutamate. When that bouton is excited, it releases those neurotransmitters, and those neurotransmitters bind to receptors that are on the postsynaptic structure. So postsynaptically here, we have in blue a dendritic spine. So this is one subcellular component of a neuron. And those receptors are located in what's shown in yellow here, which is the postsynaptic density. The way that you can, so we can, of course, directly visualize these synapses with electron microscopy. Neuroscientists want to know how synapses are functioning or dysfunctional. The technology to look at the synapse level in living human beings, so in vivo, is simply not available and will likely not be available for the foreseeable future. But there is a substantial literature in model systems which demonstrate that in vivo synaptic function is directly reflected in ultrastructural measures that are obtained ex vivo via EM. And why is this important? It's important because it might allow us to perform quantitative ultrastructural analyses in ex vivo tissue to impute something about synaptic function during that person's life. So the main question that I have is, are the ultrastructure function principles that have been identified in many different models applicable to post-mortem human brain tissue? So this is brain tissue obtained after the individual has passed away. And like I said, the real key to this and the real advance to this conceptually is that it might allow us to use EM analyses to get a window into function. And synapses are really one of those great and unique circumstances where function really is synapse, function really is structure, and structure really is function. So why would I study postmortem human brain tissue? Why would I not study brain tissue from somebody who is alive? There's a number of reasons for that. So there are 2 sources of brain tissue for research. Tissue that's been taken out from neurosurgery or postmortem human brain donations. And each one of these have their own strengths and weaknesses. The strengths of postmortem sources really aligned with my research interests, which is to understand what's dysfunctional in the brain of people with schizophrenia. So the strengths of postmortem sources is that this really is the sole source of individuals that have no neuropsychiatric disorder, or what some people might call controls in an experimental condition. So if you're having neurosurgery, by definition, there is something pathological happening that you are going in for an intervention. It is also the sole source for individuals with brain illnesses that are not treated or diagnosed by surgical interventions. So things like I'm interested in schizophrenia, autism spectrum disorder, major depressive disorder. And the other strength is that the whole brain is typically obtained. So within a single individual, you can analyze different brain regions and see if there's a relationship. However, there's the one big serious concern, which is about the potential confounding effects of postmortem biological processes on synaptic and particularly on those subsynaptic and organelle measures. And most people index this potential confounding factor using PMI, or postmortem interval. So that's the time between the individual has passed away to when their tissue is preserved. Now, there have been EM studies of postmortem human brain tissue before. I have published them myself that show that in general, the tissue can be preserved postmortem in the brain. So it's not like we have no idea as to what's going on in the brain postmortem at the EM level. However, what we don't know, and what has not been specifically done, is to do a quantitative analysis of those synaptic, subsynaptic, and organelle ultrastructural features to interrogate ex vivo whether that function-structure relationship is preserved across individual synapses. And so the data I'll present today is our attempt to begin to answer that, doing a proof of concept study. So the individual that we studied was a 62-year-old male. He died suddenly and out of hospital. He had no lifetime history of brain disorders. Postmortem interval was 6 hours, so this is about 2 hours longer than what's currently been published for FIPSIM. We did an immersion fixation of a brain region called the dorsolateral prefrontal cortex, just circled in red. Laser has a mind of its own. And this is immersion fixed, about a cubic centimeter block, 4% para, 0.2% glute. We then vibratome that block at 50 micron intervals, and those sections are then stored in cryoprotectant at -30 until it's time for histological processing. And we used the protocol that I'm sure most people here are familiar with, from Moritz Helmstadter's lab, published in 2015, which gives really great staining of membranes in tissue. That's really great for FIPSIM. So here are some of the differences, the modifications between protocols that we implemented for human tissue. So of course, the 2015 protocol, it was in mice. They used perfusion fixation, 2.5 PFA, 1.25 glute. Of course, in humans, we cannot do perfusion, so this is an immersion fixation. They used a biopsy punch that was quite a bit thicker than what we used, which was a 50 micron thick section. Of course, histology is performed immediately after the tissue is fixed and sampled. And here we performed the histology on tissue that had been stored for a little over 8 years in cryoprotectant. Helmstadter approach uses Spurs resin, which prefers an acetone as the final dehydrant. Also dehydrated at 4 degrees Celsius. We dehydrated at room temperature using EMBed 812 resin, which uses propylene oxide typically as the final dehydrant. The FIPSIM imaging was done on a Helios 5CX dual beam at the Thermo Fisher Hillsboro Nanoport in Oregon. The guys that run this were absolutely amazing to us and really generated some fantastic data. This was done using the AutoSlice and View. We did a 5 nanometer mil size gallium ion beam, 1,580 images in the Z plane. Our total volume was 2,630 cubic microns, and it took about 57 hours to collect these data. So here's a volume of postmortem human brain tissue taken at the FIPSIM. And just to orient you a little bit, in that bottom right corner is a cell body. You can see the nucleus that's right in the very corner. And then across from it is another cell body that will come in. Everything else is what's called the neuropil. This is where all of those synapses are located. This is where the communication between neurons occurs. So this is going to move through the Z plane. Those really electron-dense structures that seem to be kind of skittering through the volume as we go through it. That's myelin. That's ensheathing axons. This allows for synaptic action potentials to go down the axon much faster. So they're basically a fatty insulation. Lots of those electron-dense blobs inside of the cell body are likely to be lipofuscin. We could see some Golgi in that cell body and lots of mitochondria around. Here's the same volume, but we're just zoomed in a little bit more. So this is the cell body. We can see lots of mitochondria all around. And as we go up, we can see a number of these synapses that we're interested in. There's one here, there's one up there. So the bouton is here. All of those little circles are synaptic vesicles. It's forming a synapse onto a dendritic spine, and that dark electron-dense PSD is what lets us know that that's a glutamatergic synapse. So the first thing that we did was to complete a dense reconstruction of a subvolume, and we did this using the Amira software that is Thermo Fisher's. That's the size of the subvolume that we densely reconstructed, the dimensions, and this is the actual volume shown in panel D, and the color-coded reconstructions shown in panel E. So for example, myelinated axons were in gray, and you can see the one at the top and find it directly there in the dense reconstruction. To do this, we use the interpolate function in Amira, and this is a semi-automated approach. And the emphasis on this approach is towards the semi and less towards the automated. So essentially what you have to do is manually segment the top and the bottom of the structure. Then you segment however many planes you want in between, and the interpolate function then fills in the rest of it. So of course, more complicated structures that have awkward and complex shapes, you will have to manually reconstruct many more frames in the middle for it to actually interpolate. The pros of this is that it's not manual, which is great. You don't have to do every single slice. It's also, a pro is that it's also integrated with all of the other components of Amira, so you can use the reconstruction and analysis features that are there. The cons are that it is effortful, and it takes substantially more time than semi-automated methods where the emphasis is a little bit more on the automated, some of those we've seen today. And for a dense reconstruction, you are almost going to generate many more materials, is what they're called in Amira, than it can handle. So I think it can only handle between 200 to 300 materials. And if you're doing a dense reconstruction, you'll have 2 or 3 times that. So quantitation, we found in this dense reconstruction, this is how the cellular components were compromised, composed of the volume, and this was incredibly similar to previous studies using biopsy samples of human brain tissue. Next, we did a 3D reconstruction of the subsynaptic and organelle structures. Again, we did this in Amira, and I think this is one of the areas that Amira really shines, is reconstructing and analyzing these subsynaptic structures, like mitochondria, like the active zone, or the postsynaptic density. Here's what this looked like in sort of a presentation of, kind of looks like serial section image that you might do for TEM. These are some of the individual planes, and this is the reconstruction of a bouton and a synapse within the surrounding tissue. This also allows you to look at a PSD morphology, which can give you information about the function of that synapse. So in A, what we see in yellow is an example of a perforated synapse. You can see that little hole in the middle, and that pink structure is the bouton surrounding it. In C, we have that bouton sort of cut in cross section. We can see there's a nice globular mitochondria in red. I've reconstructed some of the synaptic vesicles in various colors, and the active zone is shown in green. And you can do really interesting things in a mirror, like these all of a sudden segmentations, and you can move through in video through the X, Y, or Z direction. So finally, quantification, what did we find? On these graphs show on the x-axis the different components of the synapse, the y-axis on the volume. And here's the real meat of the data, because we want to know, are these synaptic relationships still preserved? And the short of it is, yes, they are completely preserved in the manner you would expect based on all of the literature from model systems. We definitely have current and future segmentation goals. Again, many of these have been presented already. The segment anything in the PARI, much, much faster. Still requires cleanup, and empanada for segmenting mitochondria does a pretty good job. Again, requires some cleanup, but it's a lot easier. With that, I'll end. Thank you to my collaborators at the University of Pittsburgh, Thermo Fisher, which has been amazing in helping us get this up and running, my funding. And I also want to thank the family members who donate their loved one's brain at a very difficult time in their life. Somebody has just passed away, and they make a really generous decision to donate tissue for scientific research. Thank you.
Thanks so much for your talk. Are there any questions?
Lovely talk, thank you. So you mentioned that the interpolation was semi-automatic with emphasis on semi, and you said you were having to do manual every X slices. Give us an idea of what that X is.
So it really depends on the size of the structure. Some structures, we have one structure that spanned all 1,580 sections. Mm-hmm. And it was, of course, incredibly complex. It was this huge dendritic shaft with spines branching all around it. That one, we probably did it every 20 Z-planes, and each one is 5 nanometers. There's others, like if we were doing a mitochondria that was super globular, you could do the top, the bottom, and just one, and it would fill it right up. Mm-hmm. But it really depends on the size and the complexity of the structure. So anywhere from 1,580 to 3.
And so just to talk through that strategy a little bit, were you doing this, you would go to like a plane and you would very densely segment that entire plane? Or would you say, for this mitochondria specifically, I want to segment it and I'm going to do the 3 things needed for it only?
The latter.
Okay.
So when we were doing the dense reconstruction, we didn't reconstruct mitochondria in that, but the subcellular compartment. So like boutons, dendritic shafts, spines, things like that. We would do one structure at a time. So we would do the bottom of that spine, the top of that spine, however many in the middle, maybe 10 sections in the middle that need to be manually segmented, and then did the interpolation function.
Thank you.
It's very nice. I have some biology side question.
Yeah.
The structure preserved quite well with such brain tissue and a fixed pulse, not the perfusion as a human. So you said this one has been fixed, then the cryoprotectant. When you do the protectant means in the vibraton section, right? So I don't know, what is the condition you use for the, like a cryoprotected situation, -80, what kind of mixed buffer?
Sure, yeah. So when we're doing the vibratoming, that's just the fixed block with 0.1 molar PB filling the well, vibratoming the section. Then that gets put into a well that's filled with our cryoprotectant solution. It's 30% ethylene glycol and 30% glycerol. And so then we put the section in there, the section's fully surrounded by that cryoprotectant solution. And then we'll have like a 24 well tray, we'll fill that up. And then that gets stored at -30 for the duration of the life of the tissue. So this one was 8 years. So I collected the sample 8 years ago, cut it 8 years ago, put it in the plate 8 years ago and then popped it out of the freezer and gave this a whirl.
Great, thank you.
Thank you.
It's really nice work, and I admire your ambition. I had two questions. The first was, can you give us an estimate of the time it took you to process one volume, so we can begin to estimate the size of the total challenge?
A long time. So it, like the first presenter today, months. And so we went from something that was months, now that we're starting to do more things like with Segment Anything, things that would take us weeks to do are now done in days. So that, the more you go to automation, it really makes a huge difference. But for the densely reconstructed volume that I showed today, it probably took 2 to 3 person work hours, 2 to 3 months of person work hours to do that. And, you know, we were really super precise on this one too, cleaned up everything to where we thought it was absolutely perfect.
Great. And in the region that you chose to look, how many synapses did you find, and did you find the expected heterogeneity in structure, say, between excitatory and inhibitory?
Yeah, we did. So one of the interesting things about that came out in the quantitative data that I was very happy about is you can really see how diverse and varied the structure volumes are. And this is absolutely something that you would expect, because that is what allows, it shows the synaptic plasticity that is capable in brain, and it shows or indicates the diversity of signaling that's required in order to, you know, have an emergent property of thought. So one of the things that we were happy to see is this expected diversity in the size of the individual structures. We did see the synaptic density was about 0.4 synapses per cubic micron, and that's right around what we would expect for human tissue. There's some studies that have a bit higher density than that are in different brain areas, some a bit lower. To my knowledge, this is the only FibSim 3D reconstructed volume from human DLPFC.